cell. So that is in the case of exocytosis and endocytosis. So in a simple term, why do we need exocytosis and endocytosis? It's because we have some larger molecules that they cannot use the other means of transportation, get in or out of a cell. So such kinds of larger molecules, we have examples of them. We have saccharide. And when we said saccharide, polysaccharide means carbohydrate. And we have examples of this polysaccharide. We have number one, we have glycogen. Glycogen is an example of polysaccharide that they cannot transport using other means of transportation because they are very large, they are very huge. Like in some of this glycogen, you have them with like about 55,000 glucose. You can have 55,000, that's 55K glucose. So in one glycogen, you can have more than 55,000 glucose. So as a result of that, they cannot be transported using other means of transportation. Rather, they apply the use of either exocytosis or endocytosis. Also, it applies to proteins. Remember from basic biology, when we talk about proteins, proteins are those kinds of substances that are made they are made of an amino acid. So these kinds of huge molecules, they cannot be transported using other means of transportation. Rather, they use endocytosis and exocytosis. Okay. And then apart from this, we are now going to look at the classical demonstration of how these substances get in and out of the cell. Okay. So now, for example, look at in the case of endocytosis. This is endocytosis. So in this case, this is how some of these molecules, we call this thing as a vehicle. This structure here, we call it vehicle. This is a vehicle. This is a vehicle. So what they will do, they will just bind to these molecules. Once they bind to it, then they will now, on the surface of the plasma membrane, they will bind to it, and these vesicles will now detach itself. It will detach itself from the cell membrane and get it into the cell. So in the case of endocytosis, it's a vesicle that accommodates the molecules and then take them into the cell. That is endocytosis. And then for the exocytosis, in the case of exocytosis, look at it. This is a lipid bilayer. It's a vesicle here, and this is a vehicle, and it's made up of lipid bilayer. And also, this one is for the vehicle, and this one is the membrane. So this is the cytosol, that is the cytoplasm of a cell, and this is outside the cell. And there is also transmembrane protein at the same time. There is also peripheral proteins as well as the integral protein. So we have these proteins both on the vesicle as well as the membrane. So what actually happened is these are the molecules. These are the molecules to just be accommodated into these these vesicles, and then take them and transfer them out of the cell. So from the cytosol, the vesicle will now take the molecules, or soluble proteins. Look at them. These are the examples of the soluble proteins. They are inside the vesicles here. They are inside here. So the vesicles will just transport these soluble proteins and take them out of the cell. And that is what you call exocytosis. So ladies and gentlemen, a simple term when we said exocytosis and endocytosis is a type of transportation that usually transport larger molecules both inside and outside the cell. So for the inside of a cell, to take a larger molecules inside a cell, we call it endocytosis. While to take the molecules outside the cell, we call it exocytosis. Okay, so now So these are also more classical demonstration. Look at it. These are the food particles. So these are food particles. They are very large. So look at what happened. The food particles now bind to the membrane, and part of this membrane, look at it, it forms a vesicles. This is the cell membrane. This is the cell membrane. Look at it. This is the cell membrane. And then these are the food particles. So they want to get into this cell. So what will happen is that these protein particles will now bind to the cell membrane. Then the cell membrane will form a vesicle like this. And then the vesicles will now detach itself. It will now detach itself and get into the cell. So you see, this is in the step one. This is our step one, and this is the step two. First, the proteins will now attach, or the food particles will now attach itself to the cell membrane, and then the cell membrane will now form a vesicle. Then the vesicles will now attach itself. It will now remove itself from the membrane of the cell, and then we have something like this. So it attaches itself, it removes itself. Then look at it here, it's the protein particles. Then inside the cell, a lysosome will now break off. It will now open up, because in the lysosome we have enzymes called lysozyme. So it will break the vesicle, and now protein particles will now get into the cell. So it will now distribute in the cell. And then the next thing after this, also another vesicles will now take the unwanted, that the digestive substances that they are not longer needed. Like for example, when you digest proteins, sometimes we actually get carbon dioxide as a waste. So we don't need those kinds of particles after the digestion or after the cell is being absorbed them. So we utilize what we want, we actually metabolize them, and we have to remove those particles that we don't need them, or those particles that we don't want them. So like, for example, we don't need lactates. So now we are going to remove lactate from the cell. How do we do it? Then a digestive vehicle would also take up that particular lactate, or that unwanted materials that is not longer needed in a cell. Then it will now take it and remove it outside of the cell. So you see, this is exocytosis and elimination, because whatever we don't need in our body, we have to eliminate it through a process called excretion. So this is a typical demonstration and the explanation of endocytosis as well as exocytosis. So in the endocytosis, we are trying to get in the materials that are needed by the cell, like nutrients, or food particles like the carbohydrate and the proteins as mentioned. But in the case of exocytosis, you are removing unwanted materials, those materials that the cell don't longer need them, but they are very large, they cannot be transported using other means, then they need to be excreted or eliminated using exocytosis. So you see, this is what do you call active transport, although we actually discuss on the active transport, but very brief, but I want to also add more explanation on that, is so that at least we'll have a clear picture of all this. So now in the active transport is also the movement of substances across the cell membrane against the concentration. So in this case of active transport, it's a kind of transportation where the particles move from a region of low concentration to the region of higher concentration. That is the meaning of concentration gradient. Self-concentration gradient, it means that the particles or the substances that are crossing, or that are moving into the cell, in that situation, it is moving into a region where the particles are already there at very high concentration, that is against the concentration gradient. It means that the particles are already there at higher concentration. So you are moving a particles or molecules from the region of low concentration to the region of high concentration. So that is active transport. But one thing that we should know under this active transport, and all of us, we should put this at the back of our mind. Whenever we said active transport, there are two things that you must put it at the back of your mind whenever we talk about active transport. The first thing is the concentration gradient. The first thing is concentration gradient. Look at it. This is the concentration gradient. That is against the concentration gradient. So when we said against concentration gradient, it means that the molecules or the particles are moving from the region lower concentration to the region of higher concentration. That is one. And then second, after this is what you call energy requirement. So active transport needs energy. It needs energy. It needs ATP. And look at it. These are the ATP. So it needs ATP for it to remove the particles in and outside of a cell. So energy is needed. It's needed. It's needed. So these are the two things that we should put at the back of our mind. That in the case of active transport, we need energy is needed, and the movement is usually through a region of lower concentration to the region of higher concentration. Look at it. This is a typical example of the lipid bilayer. So now, if you look at this is a calcium. So, and in this case, this we have 1, 2, 3. We have 3 molecules of calcium here. And then here we have 1, 2, 3, 4, 5, 6. So it means that the concentration of calcium here is high. It's high. The concentration of calcium is high here. And then this is in this region, the concentration of calcium is very low. So in the active transport, the molecules are moving from the region of lower concentration to the region of higher concentration. So therefore, now by looking at this, you'll observe that the inside the cell here, or inside the cell here, the concentration of calcium is low. And outside the cell, the concentration of calcium is high. So therefore, the movement would be from outside the cell to outside the cell, but energy is needed. So from the region of low to higher concentration, and ATP is needed. This is the most important thing that you should know under transport. Movement is against the concentration gradient, and energy is required. And this energy is in form of ATP. And when we said ATP, it means adenosine triphosphate. And that is the next thing that we are going to discuss in the next maybe 10 minutes. Yeah. Okay. And then now, so as I'm saying, look at how these keys we have, these are the ligands, and this is the primary messenger, and this is the receptor. So in active transport, we need receptor proteins, because sometimes the molecules cannot just take each step into the cell. It needs to be first recognized and then bind. That is usually what happened in the case for active transport. You need a receptor, and this receptor is a molecule that usually found on the surface of the cell, that is on a cell membrane, and most of the receptor are glycoproteins or glycolipids. Most of them, they are glycoproteins. You said glycoproteins, remember from our previous classes that glycoproteins is a proteins that made up of carbohydrates and proteins, that is glycoproteins. So most of the receptors that are found on the surface of the cells, they are in the form of they are glycogen. So look at it. This is the receptor. This is a ligand. This is a ligand. First, the ligand must come recognized by the receptor and then bind to it before it's being taken into the cell. That is in this case. And then
starts from the primary messenger.
You
to
said?
the receptors.
You
Do
said?
you mean that the glyco do you mean that the glycoproteins pass from the primary messenger to the receptor?
No. The glycoproteins is the receptor. That is what I'm saying. Because most of these molecules that you are seeing in a cell, they have they have their chemical nature. Some are
Okay.
carbohydrates, some are proteins. So I'm just trying to describe receptor as a glycoproteins. And when we say
Okay.
glycoproteins, it's a protein that contain sugar.
Carbohydrate
That
and
that
protein.
carbohydrate, yeah, it's a combination of carbohydrate and proteins. That is glycoproteins. And then
Protein.
we have receptor, and most of these receptor, you see, I want you to understand something here. It's very, very important. Ladies and gentlemen, in terms of the disease that we usually come up with, like for example, most of the infectious is like coronavirus. Coronavirus needs receptor. It needs receptor before it was able to actually infect any particular living organisms. So that is why we human, we have angiotensin converting enzymes, receptor 2, which is the receptor that is required for the coronavirus to recognize host cell, bind to it, and infect that particular cell. So in our respiratory tracts, we have this receptor, which is angiotensin converting enzyme receptor. And from the virus, which is the coronavirus, it has a ligand, and the name of the ligand is spike protein. So the spike proteins is usually recognized by the receptor and then bind to it, and then the virus will actually infect the human cells. So that is how it usually happens. So it's general. It's also applied to almost every living organism, or in terms of this kind of transportation. Like in this case, we said we have a receptor. So that is always receptor ligand binding. The receptor and ligand must bind before it takes anything inside the cell. And at the same time, we have primary messenger and secondary messengers. So the primary messenger is a ligand, and the second messengers, or yeah, the second messengers usually is CAMP, cyclic adenosine monophosphates. And I'm also going to discuss about cyclic adenosine monophosphates later. So there must be a ligand receptor binding before sometimes you have these kinds of transportation to take place in a cell. So, so now look at it here. Cells must also respond to important information and filter out unimportant information. So to receive message sent by signals molecules, cell membrane contains specialized proteins called receptor proteins that bind to the signal molecules, allowing the cell to respond to the signal. So what I'm now saying is you should understand that, yes, on a cell, remember we discussed about the selective permeability nature of the cell membrane, that cell membrane allow the movement of some material while disallow the movement of some material. So how is this happening? A message must always send to the brain, must always send to the cell to actually make it to recognize, okay, which types of molecule is coming. Did I need this? If the cell doesn't need it, then it will not allow it to get in. And if the cell need it, then of course it will be recognized and then send a message. So generally there is a primary molecule, which is a ligand, bind to the receptor. And once it binds to the receptor, you have what you call receptor binding ligand. And once you have that receptor binding ligand, you need a second messengers. These second messengers, what they do usually, they amplify the signals. They send this message, they amplify like remember normal amplifier, you use amplifier so that to increase your voice so that other people will hear you that are far from where you are. So also it applies to a cell. There's a secondary messengers, what they usually